Hello everyone. So now we will move to start and perform the analysis using BRB array. And the question we posed was, which gene set or signatures are associated with breast cancer tumor recurrence? So first of all, we should open the BRB array. You just go to the BRB Array Tools, click Excel, and here you find this Excel file of the BRB array. You just double click it and it will open the Excel of BRB array. And now I have downloaded one data set, that is the excision number of that data set is there are multiple, but I am using this one, GSE 19697. Just if you want to know the details of this, you can just go here and just write this excision number, and you can find here that this data set is of gene expression signature of data from breast cancer samples of patients with pathological complete response or residual disease following the new adjuvant trial. So they discussed this in detail, and they also published this in Breast Cancer Research Treatment in 2010. So we are using this data set. This breast cancer data set contains 24 samples. All these samples are from pre-treatment breast tumor. So I just downloaded this data set here, the raw files, from clicking this HTTP, and then I stored it here, and after that I unzipped that by extracting, and now this is the raw file. So again, this raw file is further extracted. You can not this one, sorry, this one, these are 24 samples. If you just click here, you can see select all, you can see these are the 24 samples, this one, and then I extracted those samples into further unzipped those into the cell files. These cell files are actually, the cell file format are actually associated with the Affymetrix platform. So now I will use this design and perform the analysis. So let's go and jump into the start the analysis. First of all, you have to go and import. Keep in mind, you have to unzip the zipped file and also unzip each sample into one folder like I showed you. Then select the data type. I am selecting here the Affymetrix cell data, and and then I click browse, and I will go to the folder where they are saved. Here in the analysis, you can say 196, these all 24 samples. If you when you double click it, it will not show any samples, but when you click OK, right, and then you click then it will say that we have found 24 arrays in your data set. Yes, is that correct? Yes, it is correct. Then I say OK, and now there are different normalization platforms. Please select the method to analyze your Affymetrix cell files. So these are different. Most commonly used are RMA normalization method. All of these have the same purpose but different algorithms. You can see the details in the literature, but I'll just keep the default and click OK. Now this is the as I mentioned in the previous tutorial, this is the array design file. So it is saying that do you want to import yourself or use the Affymetrix probe set IDs, that is the standard. I am saying OK, use the standard Affymetrix probe set IDs. Click, and then I am clicking OK. Now it will take a bit of time because now it has to open the R, and here let's see what's now what is the project folder I want to make. I say test folder, or I can say breast cancer only. Project name should be tumor recurrence, right? Okay, this is the Excel file. I just wrote it. You can name it anyways, whatever you want. Now here it is asking for the sample description file. This sample description file is actually available here. If you see, there are different formats, say like series matrix file, minimal formatted file, soft formatted file. If here I downloaded the series matrix file, when you click this, it will open another page, and you just have to click this file and it will download. Now this is downloaded here, right? So I've already downloaded it, and here if you can see, this is the series matrix file of, and I have unzipped it. So this series matrix files contain the normalized expression already generated, and also the sample description that is available on the top of the file here. So just to make a descriptor file, this is actually the there is proper format to make a descriptor file. I'm just making it for your ease. If you go here and you just make another file like this, and here you can see that these are the all descriptions of the sample. I just copy it, and here I am just again making a transposed file like this. Now our question was, now these are the samples, these are the, in the first column you can see there are 24 samples. Now our question is to find the association of samples with tumor recurrence. Let's see where is the yeah, so here are the tumor recurrence information, right? So now we are making a descriptor file which contains the sample information and the descriptor information only. We just delete all the rest of the files, right? Now this is the sample information and the tumor recurrence information. So recurrence 0 means there is no recurrence, and recurrence 1 means most of the time it means that tumor recurrence, right? So yeah, so this is the way to make a file, and now this can be the sample descriptor file. Keep in mind you have to save it in the text format, right? Here you have to go save as and then save it in a text format, because it usually takes the text format, right? So after you save it, I have already saved it for your I just close it and I'll show you how I've made it. Here you can see that I have already made this sample descriptor file. Where is the descriptor file? Let's see. Okay, let's. So here is the. Let me make it again. Oh, why I closed that? Now this is the. No, this is something different. No, this is same, right? So let's make this file. Copy. I am copying the recurrence information and copy the sample information. Here you can see this. So now these are the samples and this is the recurrence information. So I'll save this as a text file, channel descriptor file, right? And I'm saving it in text format. Where is the text format? Here, tab delimited file. No, yeah, this is tab. Tab is delimited file I just made, and I'm saving it. Now it's saved. Here you can see this is the descriptor file. Yeah, okay. Now we going back to the. Now, yeah, now it's asking for, I do not have an experimental descriptor file. No, we have. We'll just import it from here. Where is that? This descriptor file I just made. Open. Next. Now, if it is starting to perform that RMA normalization. Now, if you see here, now it's running and it's calling all the packages of R. You don't have to worry about the R language. It will just upload all the packages like this. These all are loaded for the BRB array analysis here. Now, one thing you should keep in mind, all these packages. For the first time when you will use the BRB Array tool, it will ask for you to do you want to install this, do you want to install this, this package is missing, do you want to install it. You just have to click OK to all the packages they want to install, so that it will complete the all the process, and then after it will install all the packages, then it will perform the BRB analysis. Okay, so here I am skipping that because it will take a lot of time for all of you to go through that, because if you have any questions and if you find any errors while clicking those, you just ask a question in the question bar. I'll try to respond to all the questions. Now here you can see it's collating the experiments. All the 24 arrays are running here, and all that RMA normalization is performed on this BRB array, right? Yeah, so just wait for a while. So everything is loaded here and it's going to complete in a minute. Now it's parsing and it's almost done. Yes, now it has completed the normalization, but one thing is left, that is that in what way I want to organize, how I want to put, if I want to put any filters or anything to improve the data. For example, here it is written average the replicate spots within an array. So these replicate spots, as I showed you here in the discussion, that there are multiple gene multi-probe IDs against one gene, so it will remove those replicates and make it a uniform data. So you just click that average, usually it's better to click this average the replicate spots, and then gene filters, keep that way, everything as default, and click OK. Yeah, and now okay, now just wait a moment, it's running the file. Maybe it's not responding. Why? Yeah, we have to wait for some time. So these are few things you should keep in mind. First, that everything everything you have to install for BRB array is explained in the previous lecture, and when you will start the the processing for the first time, it will ask to install so many different packages. You just have to install everything. Okay, so now it is done. Here you can see there are 17,342 genes which pass the filtering and subsetting criteria. We said okay, that's great. Okay, now we click OK. Now it's asking for the annotation. Again, this is what I showed you here, that this probe IDs, you can see here, these are all the probe IDs here, but it's not corresponding to any genes, right? So it will if you will click this annotate the Affymetrix data, then it will annotate these probe IDs against the genes. So let's do it to make it more understandable. Now, yeah, so here you can see then the first column, let's before it gets complete, let's discuss this, that in the first column there are probe IDs which are correspond to genes, and the next columns, for example starting from B column to the S column and so on, all these columns contain different samples, and in the and these values are the log R expression values of each probe ID. Yeah, so which and these probe IDs correspond to different genes. Let's say now it's saying that writing annotation to the project book workbook, so it's loading here. Let's say when it gets done, we will see that 11% completed, so I think it will take a bit of time. 11% and then 89% is remaining. So yeah, so in the hey before it gets completed, let's see here that you can see that experiment descriptor is the first sheet contains the descriptor file. Now these are the log intensities, and then yeah, now it's, I think it's going to yeah, now here you can see that these genes are annotated. The first column contains the probe IDs, and then each probe ID contains the description and the gene excision number and the rest of the gene list, which will be published very soon. We just have to wait for a while. It's matching the gene list. You can say annotating the genes. Let's see, I think it's I hope this tutorial will help all of you to perform all the BRB analysis yourself. You can see here that you don't need a lot of expertise of programming languages and different other okay. So let's wait and wait and wait. Nobody likes to wait, but so here it's, some computers take lesser time than this and some will take more time than this. That depends on the specifications. That's why I said that all of you should have better computer processors so that you can run it fast. I think it's not a very low very up you can say difficult, because here you have run 24 transcriptome microarray data sets and you are done with it in very couple of minutes. So now there are 24 arrays in this project and the first 20 arrays are shown here. This means that let's say let's save it first. Yeah, then I'll show what it said. Yeah, now it's done. Now this is the descriptor file which we made, and now this is the annotation file of all the probe IDs against different genes, and then these are the gene IDs, this is the probe ID, this is the chromosome band, this is the number, right? So, and these are the intensities, log intensities of each probe, right? And then these are the identifiers, gene IDs. Yeah, so the first 20 are mentioned here. Like it's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So it means that the first 20 are missing, are mentioned here. You can select that, display the data by clicking display data. I'll not go into the detail because we don't have to present everything here. Our question was, again, let's go back to our question. That was, which gene set or signatures are associated with breast cancer recurrence? Let's try to find that. So if you go to Array Tool, here you go comparison between different group of arrays. For example, here we want to compare between two sets. One is the no recurrence and one is the recurrence. So we have selected this, and now if we just make it, if the threshold of it will perform the differential expression analysis and try to find the significant genes between tumor recurrence and no recurrence, and the p-value that should be significant I selected here is less than 0.01, right? So let's perform the analysis, click OK. Now it's applying the filters 100%. It's completed. Okay, everything which we are performing here will be saved in the project folder we made in the beginning, right? So that is what you should keep in mind. Now it's performing the analysis, computing gene filters. Please wait. Okay, we are waiting. So here where we saved the. Here we made the folder breast cancer, and everything is saved here. You can see it's an extra file. This file we were using. Now it's preparing to gene annotation, preparing gene tables, getting the analysis results. Everything is going to be done very soon. And now it's loading the required packages. All these packages are actually installed when I used BRB Array for the first time. So the first time you will be using, do not get confused. You will, it will take time, a lot of time, at least 1 hour or so to download everything. Yeah, now I think it's done. Hopefully, yeah, it's ready, done. So here you can see different outputs are generated. One output is this one. There's a class comparison. Here we made 2 groups. For example, if you can see here, recurrence group 18 samples and recurrence 0 18 samples, recurrence 1 6 samples. So it has compared 18 versus 6 and tried to identify differentially expressed genes between the tumor recurrence and no recurrence. In this way we can see that which genes are associated with recurrence and which are not. So based on that and based on the expression intensities, log R intensities, it's compared. Here you can see that, for example, this is the first probe set ID of this EIF3K gene, and here it's showing that the class 1, which is no recurrence, showed very high, oops, it's gone somewhere else. It's here now. When you click this, it will sort the. So here if you can see that EIF3K, here you can see that in class 1 the expression is very high compared to the class 2 of recurrence. So it means that EIF3K gene is overexpressed in class 1, that is of no recurrence, compared to the class 2, that is of recurrence. So here all the list of the genes are available. One output is this one. Others are more easier to see. That, for example, if you want to have the graphical output, here you can see, if you click here, this is the chromosomal distribution file. Then there is one interesting heat map file, this one. Here you can see it's now very clear that these are the first 18 samples with no recurrence, and now these are the first and the 6 samples of recurrence, and the dark blue represents here that higher expression, and light blue represents lower expression. So here, and all of these 24 samples are here organized. So you can see that these first set of genes showed low expression in recurrence compared to no recurrence, right? So we can say, and these last 3 genes you can see here are showing higher expression in recurrence group compared to no recurrence group. So we can say that the samples with these all samples are associated, the patients which show no recurrence of breast cancer should have the higher expression of these genes, including EIF3K, GNB4, all the genes, and these all genes will show low expression in patients with tumor recurrence. Similarly, these, for example, this UGDH gene is showing higher expression in patients with tumor recurrence compared to the patients with no recurrence, right? So I think we have answered this question, hopefully, that which gene set signatures are associated with breast cancer tumor recurrence, which you can see in this figure. And there are a lot of different ways of, and this is also a PNG format. All the formats are available, and the best thing about BRB Array is it performs all the analysis itself. It will generate different HTML files, PNG files, and you just have to open those files, and you can analyze those information based on your question. So I think this is a very beautiful picture, and we should end our presentation here, because here you can see that where is that? Here you can see that we have identified the gene set which is associated with breast cancer tumor recurrence. Thank you. Hopefully this tutorial will help you to perform your own analysis. Hopefully I will come up with more tutorials related to BRB array, because there are a lot of different other options like survivor analysis and other clustering analysis for against using those gene sets and patients data. Thank you. Allah Hafiz.